Okay, well, good morning, everyone. It's nice to be here. It's a little bit early for lectures, in my opinion, but I can't complain. We're going to talk about rule-based modeling today, and we've already had a little bit of discussion about that yesterday, Marcus, when we talked about building complex reaction networks. And so we're going to start the morning with a tutorial on BioNetGen and RuleBender, and I'm going to give it. First, it's going to start off with an introduction on rule-based modeling. What, why, and how is how I titled this. I feel somewhat disarmed because I don't have my normal laser pointer, but I don't know what happened to it. But this will do. So I'll start out with that, and then we'll have a series of short talks that kind of take us up to some of the frontiers in rule-based modeling, how we compare basically standard reaction network models that we might find in the literature or in databases, and basically turn them into rule-based models, which allows us to see inside the structure of the underlying molecules that are in the biochemical system. So Jose Tapia is going to talk to us about that. Hopefully I'll be able to talk long enough that he'll get here, because he's really not a morning person. And then another member of my lab, John Sekar, is going to talk about some new visualization methods for rule-based models, systems models, as I've called them here, showing you how to take, or basically how you can make informative diagrams that you can use to understand and analyze detailed models. So one of the big problems that one faces in complex biochemical systems is trying to translate the knowledge that you would find in the literature and previous modeling efforts into your own models and building up your own models. And nowadays, the sort of standard practice is everyone builds their own model from scratch, and it's very hard to make use of previous models in the literature. And so some of these visualization methods help you, hopefully, to get a better handle on previous models that have been developed and borrow useful things from them. And then the last short talk will be Rory Donovan, who will talk about the weighted ensemble methods for doing rare event simulations. And the idea there is oftentimes when we're using especially simulating complex systems, biochemistry, complex spatial dynamics, the timescales of those simulations are not necessarily accessible to the timescales of biological processes of interest. And so how do we make sure that using standard chemical kinetics, stochastic dynamic simulations, we can actually see the biology that we're interested in, and we can use clever sampling methods. And Rory's going to show you how he's hooked up the weighted ensemble method to both BioNetGen, stochastic simulation engine, and MCell in order to get a better understanding of some interesting and complex systems. So that'll be the first morning session up to the break. And then we're going to we'll go into the lab, and we'll do some hands-on work with RuleBender and BioNetGen, go through a series of example models. And then after that, Jose will lead you through a more complex example where we take a model that we describe in BioNetGen using compartmental description, and we integrate that with the geometry that we construct and make a model that we can simulate in CellBlender. Okay, and then that will take us up to the afternoon, and there'll be some more extended examples in the lab this afternoon, and hopefully there'll be some time to work on other advanced topics of interest to many of you to get more hands-on experience with CellBlender and BioNetGen and work up perhaps on your own projects. Okay, and all of the information for the presentations today and the tutorials is available at our wiki site, bionetgen.org. This link will take you to all the materials, including the slides that I'm presenting right now. So if you go to bionetgen.org, you'll see on the front page, I can show you briefly. Hopefully it will come right up. On the front page of, this is the main, this is the tutorial page, but on the main page of the wiki, if you just go to bionetgen.org, under news, you'll see a welcome. Here's the RBM tutorial page, and then this is the page with all the materials. It tells you how to get download and install RuleBender. You won't have to do that if you're using the computers in the labs, but if you want to put it on your own machine, you'll need to do this. One caution for people who are using Windows 8, you'll need to download the latest version of BioNetGen. So RuleBender is the GUI, and BioNetGen is the simulation engine. The analogy is cell blender. BioNetGen is to RuleBender as MCell is to cell blender. So you'll need to do that as well if you're doing that in your spare time while I'm blathering on up here. There's the slides, as I mentioned, are here. There's a quick reference guide that's sort of a quick start tutorial. There's more detailed reference material. There are a whole bunch of examples, and we'll go through some of these as time permitting in after the break, and then there'll be more examples after that. John has a visualization example that he wants to present in the lab this afternoon. And then there are a whole bunch of reference materials, articles. We have a very recently written extended tutorial that's in press, actually, and there are, I think, 8 example models that are linked to here, reviews. So lots of information, much more than we can cover in one day. Be talking about. So first things first, let's go back to rule-based modeling. And this is since you're all here, most of you have some modeling experience already. You probably hardly need the extra motivation. But I like to talk about what is a model and how does what we're doing making simulation models of biochemical systems related to standard approaches to thinking about and modeling biological systems. And so in signal transduction, of course, you see diagrams like this all the time. They show the interactions of complex biomolecules that make up the biochemistry of cell signaling and how cells respond to signals and process information. And we see diagrams like this. And normally, most biologists who are working in this area, they use such a diagram as a mental model to represent their system, and they think about this for a while, and they come up with ideas for experiments to do. So this is a cat, of course, and he's thinking about different ways to torture mice and run biochemical experiments that give us time courses of the different components in the system, the activities of different components in the system. And so really what we're trying to do is formalize that approach, and we have now a way to take this diagram to represent it formally using computer programs. This is a very smart cat who's making a BioNetGen model using RuleBender, and he's got a nice diagram of his system. But this is an executable diagram that we can use to run simulations and compute, for example, a dose-response curve. And we see here that this curve has an interesting shape that's not really intuitively obvious from the structure of the biochemical pathway here. So that there are emergent properties of biochemical systems that we can access through modeling. And then we can have a formal approach and systematically try to investigate biochemistry. So what is rule-based modeling? How does this differ from what we normally think about in terms of simple models of biochemical networks? And the basic underpinning of rule-based modeling is this abstraction. I would call this the molecular biologist's abstraction of a signaling protein. These sort of, I knew that was going to happen. Let's see. We don't want to lose the ever important audio here. Then we'll stay put. Beads on a string. So proteins can be thought of as polypeptide chains that contain conserved functional domains that interact with other functional domains. And of course, they have these folded domains that interact with each other. There are also shorter motifs that contain amino acid residues that can undergo post-translational modification. And these are the currency, the bits, basically, that get toggled back and forth in signal transduction processes. And the idea of a rule-based language is to represent the molecules at this level of abstraction. So we have sites that can bind to other sites, and we have sites that can be modified in various ways. And that allows us to construct a computational model of the interaction of these different biological molecules. So here we have one of the kinases that I've depicted, the Lin-tyrosine kinase, an important kinase in immune receptor signaling. It has a number of conserved domains, including SH3, SH2 domains. a kinase domain. It has various phosphorylation sites. These indicate particular residues, tyrosine 397 and tyrosine 508, that can be phosphorylated and modulate the activity, for example, of the kinase or of the ability of this SH2 domain to bind to other proteins, for example. And so we have now an abstraction, an abstract representation of a signaling molecule that's nonetheless quite a bit more precise and structured than just talking about an entity called Lin in our system that maybe it's active or inactive. Okay, and so we can do the same. Another example is shown here for a multi-subunit receptor. This actually has comprised of 4 polypeptide chains, one of which is disulfide linked the gamma chain. It's a gamma dimer, beta and alpha, and we represent that here as 3 different components of a molecule called Fc epsilon R1, the alpha, beta, gamma. Beta has a tyrosine that's phosphorylated. We're just representing the tyrosine and its phosphorylation state as the exemplar of the beta subunit. But we could have, for example, we could have a number of different components, for example, to represent different features of the beta subunit. So this is up to the modeler, how we abstract in detail the structural features of the protein. But the idea of the rule-based language is to provide the capability to make that abstraction. And then once we have the underlying objects in our model, the rules then are used to define the interactions of molecules. So I've shown here a standard kind of interaction. You could read, for example, a statement like this in a review article about this, or in the discussion section of a paper describing signal transduction in this system. The Lin SH2 domain binds to the phosphorylated tyrosine 218 on the beta subunit of Fc epsilon R1. And then we can precisely translate that knowledge, that information about the system, into a rule. And so the rule looks like this. It's a standard. We're using standard chemical reaction notation, reactants, arrow, products, rate law. But we have, and BioNetGen has a specific text-based syntax for that. We're going to represent the SH2 domain of Lin binding to the phosphorylated beta subunit of the receptor. On the left-hand side, so we have the requirement that the SH2 domain of Lin is involved and it's unbound. And when we represent a component in a rule without referring to a binding state or a binding state in particular, that means it must be unbound. So we need a free binding site here, a free binding site here, but this site has to be in the phosphorylated state. And then, so the reactants specify the conditions that the rule that must be satisfied by the reactants. The reactant patterns specify the conditions that must be satisfied by the reactants to undergo the transformation that's specified by the rule, which here is the formation of a bond between the SH2 domain and the beta subunit of the receptor. Okay, so we're going to construct a bond, and that's indicated by this bang notation. The first reference to a particular number, an index, refers to the starting point of the bond, and then the end point of the bond is marked by the second reference to that number. So each bond is represented by a distinct number in this representation. We can have multiple bonds in this in the complex, but here we're just indicating that a single bond is being formed between these two sites, and then these two parameters are the parameters that are going to govern the rate of this, the transformation specified by this rule. They are the constants or the numbers that go in front of the mass action terms for this reaction. So the total rate of the reaction, which is given down here, is going to be, for example, in the forward direction, KPL. So, and I'm kind of using a little bit vague words or a little bit general words to instead of saying that this is a constant, I keep saying that this is a number or a function, because in fact, although many times when we represent these systems at their most basic level, this is a constant number, we can also make this a function in BioNetGen. So this can be an arbitrary function of anything in the system. So any reaction rate is governed by this function or constant, times the concentration of each of the reactant terms. So here it's the concentration of free SH2 site, free SH2 components on Lin molecules and free phosphorylated beta subunits on receptor molecules. So the standard mass action appearance of the term is true, it holds if this is a constant, but this can again be an arbitrary function. And I'm mentioning, just trying to plant that seed in your brain now so that when we do different kinds of rate laws in some of the examples later on, it won't seem strange that I'm going to put the dependence on other properties into this term. But for now, we can just think of this as mass action kinetics, and this is a constant. And so a couple of other key features about this rule. The components that we've specified here that are interacting are called the reaction center. So those are the elements that are directly modified by the action of this rule. So these two components are going to be bound to each other when this rule acts on them. And the context is everything else that's specified in the rule that's a condition for the rule to generate this reaction, this process, but that are not modified by the action of the rule. So here we have a single element of context, which is the fact that this beta subunit is in the phosphorylated state. That's not changed by the binding reaction that's carried out here, but it is necessary for the bond to form. So that's what we call context. And the key thing, basically all of the rule-based concept is embedded in this last statement, that everything else we don't care about. We only specify the things that are necessary for the rule to happen. We don't specify what else can be going on with these molecules. So we don't write, we don't care. So anything else that's not mentioned here doesn't affect whether the rule can occur or not. So for example, the Lin tyrosine kinase has other domains. It has other phosphorylation sites. In principle, those could modulate the behavior of the Lin molecule. This rule says that those things do not affect the rate at which this reaction happens. All I require is that Lin has an unbound SH2 domain. Frequently, we don't have information about the interactions, how modulation of the activity of one domain affects the activity of another domain in proteins, although undoubtedly those are important in many biological processes. The kinds of measurements that we have available, the measurement technologies that we have available, don't allow us to easily extract those quantities. And so sort of following a principle that says, well, if we don't know, we can make, we want to make the most general assumption, we only write down what we absolutely know. If we know that there's additional context, we can clearly add that into these rules. We won't actually talk about it much in the workshop, but one of the key areas of research is if we have experimental data, can we extract information about molecular context from that information by fitting models to data? Can we get the parameters that govern the interactions? And that's an ongoing research area. Okay. So why do we need this? I mean, I think I've given motivation for the fact that we know that we know about the structure of molecules that are involved in signaling biochemistry, and so the simple answer is that this is what we need to represent that biochemistry. But why do we need a different set? Why can't we just write down A plus B goes to C for the molecules that we're interested in and leave it at that? And the key point here is that these protein domains are multifunctional. There are many different things that can happen at one time. There are many potential interactions between those sites. And what one finds very quickly if you try to write down the models of these systems, you can write a very simple set of rules describing a simple set of molecular interactions, and that generates a huge amount of complexity. So when you start to try to figure out how many different combinations are there of these 4 different signaling molecules that are involved in the first steps of the immunoreceptor signaling, in the case of the IgE receptor, in the early days we wrote down a model of this system, and it had 354 different possible complexes that could form if we considered only dimers of receptors. And if we had ligands that made aggregates larger than dimers, we started to get into thousands or hundreds of thousands of possible species. And so very early on, we discovered that if we wanted to make effective models of these systems, we needed to have a computer do some of the bookkeeping for us to keep track of all the different possible species. And we needed to have a concise way to represent these interactions and figure out what the possible species were. So we need to have algorithms that could generate those for us. And we'll talk a little bit about that as we go along. So how does this play out? So let's take an example of a receptor that's involved in another signal transduction system. The epidermal growth factor receptor is depicted here. So we have here, and I'm showing a different representation of this system now. And the idea is that I'm showing the different molecules that are at play in early signaling events in this particular pathway, the epidermal growth factor receptor signaling pathway. And I'm showing just how they can interact with each other and what sites can become modified. We call this a contact map. So it's a concise visual representation of the molecules and interactions that are present in our system. We have the ligand, we have the receptor, we have a couple of adapter molecules, Schick and Grab2, and we have the first real effector molecule in this system, which is SOS, which acts as a guanine nucleotide exchange factor to activate RAS. And so, which is not even part of this initial model of this system. And so what's shown here, the receptor and the ligand can bind. There's a dimerization site on the receptor. There are two conserved tyrosine residues that become phosphorylated upon receptor dimerization. And upon phosphorylation, these sites can bind to the Grab2 adapter or the Schick adapter. Grab2 binds both Schick and the receptor, so there's competition for binding at this site. And the Grab2 also binds to SOS. So very small number of interactions, fairly simple system. You could write out in a small paragraph describing the interactions among these proteins. And these have all been well known for probably about 20 years now, these interactions. But the complexity is still pretty problematic for the system. So if we just do a simple accounting, looking at the tree of the possible states that can arise here, at each site, the ligand, for example, the ligand can be bound or unbound. For the phosphorylation site, we can have it be unphosphorylated, phosphorylated. If it's phosphorylated, it can be bound or unbound. If it's bound, it can be bound to another protein or not bound, and so forth. And if we do all of this accounting out, we see that for the numbers here represent the possible states at each site. We multiply those together, and that gives us the number of possible monomer states. So there are 48 different possible states of the monomer here, which require, when we're doing the monomer accounting, we're assuming that the dimerization site is not bound. Okay. Then when we try to figure out the number of states of the dimer, then we have this 48 on one side. If we account for symmetry, the total number of dimer states is number of monomer states times number of monomer states plus 1 over 2, which is 1,176 possible species. So this is just a simple example, or not so simple example, I guess, to show you how the counting really explodes on you. As soon as you start to put in a few interactions, you get this combinatorial explosion, call this combinatorial complexity. And it makes it very difficult if you had to keep track of all of these in a model, or in your standard if you had to enter these in the M-cell reaction window, it would be a little tedious at best. And you probably couldn't write down 1,176 reactions without making a mistake. So the funny story about this was when we first started making the model of the IgE receptor system, it was about 2001, and I came to Los Alamos where this work was going on. As a postdoc, there was a committee of, at that time, 4 scientists, and I was the 5th that was added to that group. And the way that we were writing this model was to divide up the 356 different species, or 354 different species among us, and write all the reactions for each one. And then every week we would meet and discuss the reactions that we had come up with and argue about whether there should or should not be a factor of 2 in front of this reaction or that reaction. And the problem was that this process didn't converge. And so some of us got impatient and started writing computer programs to do it. And those were more accurate than we could do on pieces of paper. Yep.
Hold that thought a second. We had a way of writing down a simplified version of the K2 reaction that was talked about here. And I had Python code that generated the reactions using MCell. And this is a simplified version of it. The MDL file is for the reaction is 1.2 gigabytes. And there are 28 million lines of reaction. And MCell parses it in about 30 seconds or so. MCell doesn't care
Yeah.
that it's that big. And it doesn't slow down anything, but I doubt that it's correct, even with the code that generates it and everything. So,
Right.
yeah, there are problems.
Right. So everybody's very quiet this morning. So is all of this making sense? Does anyone have any questions about it so far?
One question I have, not knowing anything about chemistry, is that if you have millions of lines of reactions, then you have millions of lines of numbers that you have to know, which are all the.
So that's not necessarily true, because it would be true if we knew all the context, right? If we knew how every site interacts of a given protein interacts every other site of the protein. But when we look at this kind of a representation of the interactions in the biochemical network, basically we have parameters describing each of these interactions, right? And we have parameters describing, for example, the post-translational modifications that can occur. And if we were more careful in our accounting, we would have enzyme-substrate interactions that we considered, and those would represent parameters. So typically, for every pair of molecules in our system, we're going to have a handful of parameters. And in the case of well-studied systems like the EGFR system, there are measurements of most of these parameters available. And moreover, we know the basic biophysical chemistry of those interactions and have sort of pretty reasonable handle within an order of magnitude or so of the values of the parameters that govern their interactions. So it's not like we're going at this completely blind and that we have to consider every possible combination of, every possible state here has a different propensity to interact with every other possible species in the system. There's really a fairly limited number of parameters. And in fact, if we have decent experimental kinetic data, we can infer many of those parameters directly from the experimental data. And moreover, if you wrote down any model that considered these interactions, but even ones that don't account for all of this complexity, you still have to make some guess as to what the parameters are that govern each of these. So this picture, this map, tells you what a minimal model of this system has to look like, right? And it's the same, and the parameters for that minimal model are more or less the same parameters of this larger model, at least as a starting point. So we don't have to. So Tom doesn't have to know 2 million parameters to have 2 million reactions. He might only have to have 30 parameters, and those 30 interactions that he knows generate this complexity. It's very much like the situation in the protein world. Proteins form very complex three-dimensional structures. What we know about protein-protein interactions is mostly consists of decomposing those interactions into the possible pairs of amino acids or possible pairs of atoms that make up those amino acids and writing an interaction energy function in terms of those pairs, right? And then we can describe the full interactions, at least in an approximate way, in terms of that pairwise decomposition. That's very much like what we're starting to do here. It's just that people don't usually think about it this way, but this is a good way to think about the problem. Okay, so let me just summarize what we've really talked about so far. So why use rule-based modeling? Well, if we want to model complex biochemistry, these rules basically represent the simplest way to describe the interactions in a biomolecular system. So they're simple. In this contact map representation, we're basically representing the context-free interactions of these molecules. So it's concise and flexible. The flexibility comes to, so rules can be obviously, as we add more and more, we can add more and more context into rules as we have more information and build up more and more complex models. We're going to talk about shortly the flexibility that when we specify this model in terms of rules, we're not specifying yet a way of simulating the model. We're not specifying, for example, whether our model description of this system is going to be deterministic or stochastic. We're not saying whether it's well-mixed or spatial, and so forth. So we're flexible with respect to the ways that we can simulate models that are specified using rules. And so it's great for interacting with a system like MCell. We can write a rule-based model in terms of biomolecular interactions. We can perform preliminary simulations of that model using deterministic, approximate, well-mixed assumptions about the reactants in the system. And then we can go on and using some of the tools that we'll talk about today to test the limits of those assumptions. We can first look at does the inherent stochasticity and discreteness of molecules that are involved in these processes affect the dynamics. So if we had our Latka-Volterra, first question is, so I make the differential equations for Latka-Volterra. I do a simple simulation. I see the predator and prey oscillating up and down. Then I say, well, if I only have 100 wolves and 100 sheep, and I do a simulation stochastically, what happens? And we see that sometimes all the wolves can die off, and then the sheep grow exponentially. And I can see that because my simulator crashes, takes over all the memory on my system, and so forth. Or maybe all the prey dies, and then everything dies, right? And so those limits can be sort of outlying cases can be observed in stochastic simulations. And then we saw yesterday really beautifully what happens when we make such a system spatial and stochastic. We get these crazy explosions like fireworks on our screen showing predator and prey going up and down. So from the same set of interactions, we get all these different kinds of phenomena that can occur in our system, some of which we don't really even have a good theoretical handle on what's going on with them. OK, so that's about simulation. On the biochemistry side of things, and I mentioned that an existing problem is we have these very complex signal transduction cascades. They're mapped out. We have diagrams that are in lots of review articles. But we really don't know how, when we put all of those complex interactions together, we get the emergent features of biochemical signaling networks. And we'll talk about a few examples as we go along. And the goal is, perhaps rules can be a flexible, precise, concise way to represent these interactions, and then allow for the development of reusable libraries of signaling interactions, such that when you want to make a model of your T-cell receptor signaling cascade, or EGFR, or TGF-beta, or whatever it is, you can go to a database and pull out the set of interactions that are of interest to you, and allow you to make, for example, a comparison between the model that you're developing and the experiments that you or your collaborators are performing. And then we'll see some examples today, too, of how one of the aspects of rules that really plays into the possibility of reusability and libraries is the ability to visualize them, so we can use our visual processing systems to get a quick understanding of what's in a model, rather than having to sift through pages and pages of equations, a plus or minus sign on any one of those pages, which can change the meaning of the model entirely. Okay. So an example of what I mean by this is trying to compare two different models in the literature. So what I don't have on this slide is the fact that we have two complex models that involve parts of the MAP kinase signaling pathway, and those are shown in green here, actually. But in general, we have a large number of molecules, complexes, and molecular interactions. And if we go to the biomodels database, and I would encourage any of you to go on the web now or sometime later and look up these biomodels 543 and biomodels 19 and see if you can come up with any information about those models that's anywhere nearly as compact and accessible as the visual representations that I'm showing you here. So this is kind of a plug for the tool that Jose is going to talk about in the next talk. We were actually able to go to these models, which are specified in terms of reaction networks, and extract exactly what different interactions are implicit in that system, and then provide a visual representation. So the ability to look at these models in terms as a set of rules, a set of structured molecules and interactions that are governed by rules, enables us to quickly make a comparison. And we can see that many of the components of the two models are shared, but their connection to for example, the receptor aspect of the model is quite different. So here we have a JAK kinase that we have interferon gamma interacting with the receptor. IFN is representing interferon gamma, and a JAK stat signaling cascade, but has a connection to MAP kinase. And here we have the EGFR connection to the MAP kinase cascade. So the ability to visualize, I think, and think about molecular networks in terms of protein-protein interactions, I think is very important and a great benefit of our approach. Did you have a point?
I was just wondering what, if we look at components that are in common, like the MEK, ERK,
Mm-hmm.
with the two phosphorylation sites.
Right. So so you're getting at the fact that
which are really the same.
Yeah,
The same molecule in both models. Right?
they are. They're represented. There's slight differences in their
Looks like there's something
representations.
Looks like this one has more.
They just haven't been ordered, actually.
Yeah, but this, but the one on biomodel 19 might have a richer, more complete representation of MEK and ERK because they have a little bit extra. Like if you look at ERK.
There's 6 versus 5. I'm parsing the difference there. It's because in this, it's actually this component here. It's the internalization. This is representing the internalization. So somehow in this model, the MEK is allowed to internalize when it's a part of the receptor complex, goes into endosomes, and that could allow for signaling from those from the internalized MEKs. It can also allow it could also be a way of allowing for degradation of this molecule, and we'd have to look into more detail to see how that's.
A little bit richer.
It's a little bit, yeah.
MEK. So then you could maybe start thinking about how if you were going to make a composite model.
Right.
It's putting aside.
Jose will talk a bit more about this in the next talk, and I think we're actively developing ways to even compress the information that's presented here. So the number of, there's actually additional numbers of, there are additional components here that are not, you can group them in certain ways to make the logic of the model even clearer than it is in this representation. That's something that we're actively working on, actually. You have a question?
Yeah, so I'm still a little bit nervous about what you said about rate constant and
Mm-hmm.
how it is not such a challenge. Because at least maybe for this system, it's not such a challenge,
Mm-hmm.
but in other systems, there are a lot of isoforms for different species. They have different reactivities. And for me, that's been probably one of the biggest challenges of building these sorts of models. So I'm just wondering, how do you handle scanning the literature? It takes a long time.
Right.
How sensitive have you found your models to be different reaction rates and stuff like that?
Right. So I don't mean to suggest that parameterization is not an issue for these models. It certainly is. And what you're saying, I mean, all of these molecules, I mean, it's a gross oversimplification to represent any of the molecules that's shown on this diagram as one thing, right? They have splice variants. They have different isoforms. And all of those things are actually actively regulated in signal transduction, something that we don't talk about very often, but is undoubtedly true. That in a cell and in two different states of a cell, the functionality of proteins can actually be regulated. So, and what are the parameters that govern that? How do we get it? And yeah, we don't know. And you're not going to, you can read the literature for a long time and you will not find the answers to those questions. So I'm not saying that we know that we have answers to all of those questions. What I would say is that we have a systematic way to write down the effects of those changes. And I would also say that our experience is that in many cases, you can have a model which has a large number of parameters, but if you have some data to constrain the model at multiple levels, so you have measurements of multiple different signaling molecules that are part of this cascade, and of their kinetics, for example, you can constrain a lot of the parameters pretty effectively. And combining that with sort of known constraints, make models that that have reasonable behavior and make some interesting predictions that you could go and test. Yeah.
Do you also use those sorts of constraints to figure out like where you can cut off a signaling network? Like in terms of, like there's so much feedback in these networks, how do you know?
You don't, yeah. The feedback is critical, I think, too. And increasingly, we realize that's the understanding of what's going on. Is what are the positive and negative feedbacks, and which ones are important in this given biological context, which we have to basically find by doing working closely with experimentalists to understand those.
A few
Yeah.
years back, John Guggenheimer gave a really scary talk about analysis of dynamical systems, saying that you really can't cut off a network anywhere.
No, probably not.
And it's scary.
But you can hope that timescales do save you a little bit. So, for example,
Make a time scale.
the receptors when you provide a stimulus to a cell, in the first 10 minutes, that cell hasn't had a chance to remodel the expression of all of the proteins that are involved in the processing of that signal. We hope, and so it's still behaving sort of in a more or less a way that's more or less consistent with our model of how it should behave. But 5 hours later, it may not be. And so we have to be aware that that's a limitation. And there are all kinds of feedbacks that are occurring at the level of 5 hours: splice variation, transcription, translation. All kinds of regulation, some of which we don't know about yet, are happening and are going to change the behavior in that time scale.
At the end of his talk, and Mill was like, that was kind of the heuristic that he proposed to time scale.
So is some error analysis part of this process? That these are all interdependent, right? Yesterday we had trouble finding 3 parameters that would actually be stable enough for all C's. Now you have, let's say, tens of them, and this matrix of parameters, if you disturb any of them, it propagates, right? Like the error propagates. So
They're usually compensate. So what we find typically, and I haven't I haven't put that into this talk, but we do consider how do you estimate the parameters for this system. And what we find is that you can find many different parameterizations of the model that are consistent with a given set of data, but there are definite constraints on those parameters. So not every possible combination, and not even a very high proportion of the possible combinations are feasible, but there's still a space of parameters, right? If I had 2 parameters, there would be some region, of non-zero area, not a single point of feasible parameterizations of the model that would be consistent with what I know, statistically significant, consistent with what I know. And then, within that, so if they then sample the model within that space of those parameters, I can say all of these possible predicted behaviors are consistent with what I already know. And I have to go out and make some measurements of those predictions, and maybe that can help me further. If I make the right ones, I can further constrain. Maybe only a subset of those parameter sets are consistent with the data. And that's the general approach that one needs to take to these problems. So the idea, every experiment has a reward potentially for making improving my understanding of what's going on in the system. And I have a way of tracking that by making models instead of just mentally tracking it, which can be have problems, especially when there's feedback. So okay, we have some nuts and bolts to get through, so we should probably go on and do this. What did we schedule? We scheduled this for 45 minutes, right? So I'm already over the time. But we'll be okay, I think. We have. Well, I could turn it over to Jose, but he's not here. So anyway, let's talk. So I do want to say, I mean, I don't want to give the wrong impression that BioNetGen and the tools that and RuleBender, the tools that I'll be presenting today, are the only rule-based modeling tools that are out there. There are actually quite a lot. This has become a very active area. So there are the BioNetGen tools, one of which is also it has been developed by former colleague and collaborator and co-developer of BioNetGen, Michael Blinoff at University of Connecticut. And the Virtual Cell project has integrated within it an interface to BioNetGen and rule-based modeling, which is very nice. And then on the other side of, in terms of other languages, the Kappa language is a very close cousin of BioNetGen, and there are a number of simulation tools related to Kappa that you can find on the web as well. Simune is a tool that lets you build rule-based spatial and spatial models and simulate them using a completely graphical interface, which has its pros and cons. So to make complex models in the graphical interface, it takes a considerable amount of time to craft them, and at the same time, they're intuitive and in a lot of ways, the ways of constructing interactions. Whereas in BioNetGen and RuleBender, we're gonna have to write code. I'm going to give you lots of code examples and hopefully simplify that process. But so that we really have two different approaches at this point to how models are specified. Yet another way of approaching the problem is encapsulated in this PySB framework, which is a Python framework for building essentially rule-based models. PySB is actually built on top of BioNetGen, so it uses BioNetGen as a model compiler, but you write your models in a sort of variant of Python, customized Python language. That's the PySB language, and what it really gives you is once you understand the basic concepts of rule-based modeling that we're going to talk about in the tutorials today, PySB is a great way to build structure on top of that. So we have, whereas in BioNetGen, we write down rules for every single kinds of combinations of molecules we have in our system. In PySB, we can write down rules about rules, and so build it at a higher level, and that's great. But this is entirely a programming-based approach to models, which is has its pros and cons again. And then finally, all of these tools we're working on ways to connect all of these tools together, and one of them is this SBML multi-framework, which is a standard for encoding rule-based models, which is just nearing its final stages of approval as an SBML standard. And so in the future, we'll be able to exchange models among the different frameworks that are described here, which is a good thing, I think. And I would argue that the multi-way of representing models is actually the most general way of representing biochemical models. And so ultimately, this should become the core of the standard. But that's maybe not.
Space?
Huh?
Does it have space?
Not. There's space SBML level 3, and we'll hear about that from Devin. At least we'll experience it from Devin at some point in the workshop. There's an SBML level 3 spatial as well, and they can be combined, and we're the first people to combine them in a systematic way. They're neither combine proposed but not implemented standards, but we actually have implemented to some extent both things. Okay. So maybe this is a natural breaking point because I'm going to go from here into more of the nuts and bolts of how we actually write rules and interactions. So why don't I stop and we started at 8:30. So now just take questions. Does anyone have questions so far? Of course, the alternative is that since Jose just walked into the room, he could just start immediately, since he's ready to go, I'm sure. It's okay. We're. Yeah.
You set an example of this classical sort of force field analogy that people use to build up these protein-protein interactions. So they derive the parameters from some different experimental values, and they use perhaps the most
Right. No, we don't. Let me, since this has come up, I'm a little bit worried about opening a can of worms, but I have a whole presentation about this, and I'll show you a little bit of what I have in mind, if I can remember what it was called. Did I? Let's see. So the question was asked about, just in general, about how do we estimate the parameters. And I gave a talk not too long ago about work which tries to address this issue using so-called Bayesian parameter estimation, that embodies what I talked about, this idea of using Monte Carlo sampling to find these feasible sets of parameters in the parameter space of the system that's consistent with data. And we use some basic machine learning approaches that come from machine learning to do that. And I like cats, and I talk about cats when I can. And the key part of it, which is going to address your question, I'll jump to here, is the idea of decomposing the energy. So the energy of the formation of a molecular complex in terms of the pairwise interactions and then higher order interactions. So here, the energy of binding of multiple molecules to a scaffold is the sum of the individual binding site energies plus the energy that's required to make a cooperative interaction here. And so the closest analogy I can come up to combining rules is the idea of cooperativity in interactions. So, we typically can get from various kinds of biochemical experiments, these are free energies of interaction between proteins, right? And these can be measured directly through experiments like surface plasmon resonance, BIACOR, for example, where we take usually pieces of proteins and allow them to interact with each other and have sensitive measures for when they're bound, and use that to characterize their binding affinities and reaction constants. And if we're really clever in how we design the experiment, sometimes we can get at cooperative interactions between those. But most often, we don't know about these terms. And so the goal of this particular work, and it's not published, unfortunately, was to try to infer these interactions from biochemical data. So we have our model of EGFR system. So this was a real working model that we were using. And we parameterized it in terms of binding interactions and then higher order interactions. And it turns out that we were comparing the results that we were getting with a previous model that had been developed of the system. And both of the models had all of these interactions except one. In the previous model, the model of Holodenko and his collaborators, they had removed this term. They had basically set this interaction energy to be infinity, meaning that you could not have a receptor complex which is bound to Grab2 and Schick at the same time. So this is a very standard kind of assumption to make. So we have a receptor. It has we know that it has many phosphorylation sites, that those phosphorylation sites all become phosphorylated rapidly when we put ligand into the system. But it's a pain in the butt to have to write down all of the complexes involving these interactions. And so let's just assume for first approximation that we don't have any of these complexes. And that's completely equivalent to setting the energy of this complex to be infinity so that we never would find it in a real system. And it limits the number of biochemical species that I have to consider in the model. If I have all of these, if I allow all of this, I have 350 reactions, 350 species. And if I get rid of that one term, I get 20. I think I get 18. Okay, and so if I have to write, I have to write the equations, and you'll see there's typical model in the literature has a table. In one line of the table is one reaction of that model, which if you allow that term, it turns into 3,000 lines.
So in a sense, the energy we're talking about is human energy.
Yeah, exactly.
Write all that down and think about it.
So in this study, we asked the question, well, what's the consequence of not considering this interaction? And it's interesting because so what's fun about this is that it actually mattered in this study, and I'm not going to go through all the mechanics of this, but we have energy by next-gen language is sort of an advanced topic. It's beyond the scope of the workshop, but we have information about that on the wiki, on the BioNetGen wiki that I showed you. And if people are interested, I can point you to the correct links on that. It's also pretty clear from the documentation. So we did this parameter inference where we expanded the energies of this model, the parameters of this model in terms of binding terms and cooperativity terms. With the default assumption, we applied this lasso regularization. We assume that the cooperativity is 0, so we get rid of this term unless we need it. The fitting procedure finds that we need it to fit the data well. Okay, so this lasso. The idea of the lasso is to say we're going to squeeze out the complexities in our free energy parameterization unless we really need them. And so what that looks something like here is an example of represents a parameter. So on the x value is the value. The x-axis is the value of a particular parameter. The red indicates the probability of our prior distribution. So this parameter we're assuming that it should be centered around 0, and we're only allowing it to move away from 0 if there's strong evidence to support that. And what we find when we fit this Holodenko model to the data is that we're pushed away, and this cooperativity parameter has a non-zero positive value, and we need that in order to be able to fit the data. Okay, so what we find building these models, these are details about the actual models that are used. So we can fit the data pretty well with both. So this was the model. This is the 350 equation model, and this is the 30 equation model. And both of them sort of fit the data pretty well. This one fits slightly better because we've actually allowed this model to have more parameters. So this is a case where the rule-based model has fewer parameters because of the way we built the model than the original biochemical model. So this had, I think 25 reactions, and this model has several thousand reactions, and yet this model has more parameters. And as a result, the Holodenko model actually fits the data slightly better, whereas so the mean of the log likelihood is somewhat lower, but because there are more parameters, there's a penalty. So the models basically fit the data about equally well according to Bayes' criterion. And what's interesting is, okay, so the punchline and the long-winded answer to the question, I just thought it was good to show you kind of how we approach the parameter estimation problem in general, since people were interested in that. We find two different mechanisms. So when we assume that there's, that free energy, that we can't make the two adapter proteins bind to the receptor simultaneously, we get one mechanism, and that mechanism is is embodied by this distribution. It's basically a positive free energy term, which is an unfavorable interaction associated with phosphorylating this adapter protein Schick, and it's binding to the receptor. So what happens is Schick, the receptor itself is a tyrosine kinase. It phosphorylates Schick. The result of this phosphorylation is to weaken this bond and causes Schick to dissociate from the receptor. So that's the model. And the goal of this, the data shows strongly that there's a transient activation complex, and then that complex dissociates, it goes away. The level of phosphorylation of the receptor and the level of activated receptor complexes goes down after a brief transient activation period. And so this is a mechanism that you can get transient signaling with this. So you have, the Schick, so the receptor is initially unbound, unphosphorylated. It becomes phosphorylated. Schick binds. It gets phosphorylated. Boom, it breaks the bond. So we have a transient period of signaling. Okay, so that's one mechanism. But we were able to fit the data without this mechanism at all. And so in the case of the full model, the energy model that we developed, we find something else. We find a mechanism that was ruled out by Holodenko because they didn't consider any of these species, which is that if there's a positive interaction between the two adapter molecules, a positive free energy of interaction on the complex, we can also get a switch-like signaling behavior. So here we see that the cooperative free energy GRAB2 receptor SHIC is negative for almost all of the parameter sets that reasonably fit the data, well away from our prior expectation of the 0 value. And that's embodied by this positively cooperative complex. So it's exactly the opposite of what Holodenko was assuming in the model, that one way to fit the data is to have a positively cooperative complex where these states are preferentially formed. And somehow then this leads to switching off of the signal. And there's the mechanism for that is shown here, that when the SHIC gets phosphorylated, it essentially, remember I said there's competition for the SH2 domain of GRAB2, so the SHIC, the phosphorylated SHIC, can pull GRAB2 onto the into a complex with SHIC, and that weakens the interaction with the receptor, and the loss of the cooperative binding partner for SHIC weakens the interaction of SHIC, the formation of SHIC complexes with the receptor overall. And so it's not the most intuitive thing, and it's certainly not something that you would have just immediately thought of prior to seeing the result from the parameter estimation, but it sort of pointed the way to getting at a mechanism that we hadn't considered before. So I don't know, I can't tell you today which one is correct. I can say that basic structural considerations don't rule out this complex. So, and I think in many, many cases of receptor signaling, it's possible for many proteins to come together and form a single complex. So this is a structure showing a region of the disordered cytoplasmic tail of the receptor interacting with three different signaling, with three different adapter molecules, and showing that they can all essentially bind at the same time without creating steric clashes between them. Does that mean I know that they do bind all at the same? No, I don't know if that's possible, or that if it's energetically favorable, but I can't rule it out a priori. So hopefully that was it's a good story. So it tells you something about how we approach parameter estimation. It tells you that we can often it's worth to look back just at the results here. What's shown on so here's the data. These are you can see the amounts of data and the number of number of different data points that we have, kinds of observations that we have that we use to constrain the model. And so with it's not surprising that with on the order of 30 to 50 parameters, we should be able to fit the model data well. But what's shown here on the last panel is kind of interesting from the point of view of can we learn anything? So you could say, well, you should, if it's true that you can, that if you have so many parameters you can fit anything, then you shouldn't be able to get any kind of precision in the predictions out of the model because, there must be, an infinite number of combinations that give agreement here, but you could get all kinds of crazy behaviors in something that you didn't observe. And what we find is that that's not true. So that the output of this signaling cascade is the recruitment of this SOS protein to the membrane. And we see actually that we get reasonably tight predictions on, for example, how much sauce is recruited to the membrane as a function of time and dose of EGFR. So that the different colors represent different doses of ligand. So we can, we see different properties of these curves. So if we went and made measurements of the kinetics of sauce recruitment, and we, as a function of dose, we would probably that would probably put quite a few additional constraints on the model based on the different behaviors of these curves as we change the ligand dose. So it's very likely that if you had a musical soundtrack going, it's very likely that experiments would which would make the model struggle to fit this data, We probably, yeah, that's just a guess. I haven't done it.
But what do you make in the Kolodenko model, the fact that it seems to be fitting those 4 measurement points slightly better than?
Yeah, so here, yeah, this is like the most disturbing thing in the thing. It's not clear. So one of the most difficult things to estimate from the experimental data is what are the errors on the measurements.
Yeah.
And so, I mean, depending on what errors you believe are present in the data, you will or will not think that the features that Holodenko model is obtaining, are reasonable. And we really just don't know. This is the only data we have. So you've got to go and do more experiments, clearly, to help to resolve that issue. And there clearly, even with all the parameters in these models, there are clearly discrepancies between the data and the models. So it's simply not true that you can just take any complicated model of these systems and parameterize them and find that they agree with the experiments. And it's quite the opposite, is that you really have to work very hard to get models to fit the data.
So, well, and on that point, we had the good fortune of having Francis Crick at the Salk Institute for a long time, and he would come visit the lab a lot. And during one day, we were having a discussion about modeling and what should models be doing. And at one point he said, well, you shouldn't have a model that fits all the data because not all the data are correct. Which is actually, very insightful.
It's something that we struggle with too. You have your collaborator, he goes, and he insists that those are just really clean westerns, and that he didn't make any mistakes in loading any of the lanes, and this is what we get. And we get the the signals on at 10 minutes, off at 20 minutes, on at 30 minutes. Do we fit that or not? Because clearly, if I put that if I run that through parameter estimation, that's going to invoke very strong constraints on my model parameters. In fact, I probably have to change my model quite a bit to make it behave that way. So, yeah, it imposes. Yes, you have deep questions that have to be answered. I feel like in future versions of the workshop, we should have a whole session devoted to all the practical issues of that. Here we're focused more on the mechanics of how do I make a model of system X, but in reality, once you got a handle on that, you're going to face all kinds of other questions that are actually harder to address.
And I could tell a story maybe later about how Joe and I made our model was making prediction about the behavior of the neuromuscular junction that mostly fit our experimental data, but there was one parameter that the model just wouldn't line up with what we saw in the experiment. And then finally Joel figured out that we had a stray capacitance in the input amplifier, the current injection amplifier, actually, that we weren't compensated for. And we went back, Joel went back and tweaked the circuit, and we did the experiments again. And everything came out the same, except for that one number in the measurement, and then it agreed exactly with the model. So that's a really fun example.
How's everyone doing with caffeination and sugar levels, blood sugar? Good. We can take a break, or we can keep going. I want to tell you something about the nuts and bolts of building models and simulating them, but I can go either way. How are you guys doing? You're good. Okay. You're not dying. You're not like, okay, enough already. Okay. So yeah, hopefully I'll try to do this in a reasonably efficient way, and then we can take a break and hear about more advanced cool stuff. So basic process of modeling, and many of you are experts on this, but we want to figure out first, we have a process of interest. First thing to do, what components are we going to put in our process? What molecules are we going to model? What interactions among those molecules, and so forth? So we've seen that rules allow you to write those down in a concise way. So we translate the different molecules into structured objects, and we translate the different biochemical interactions into rules, and we'll talk about the nuts and bolts of that in this segment. And we need parameters, and we've talked already about parameters, things like the initial concentrations of the molecules and the rate constants that govern those interactions. And we've seen that you can actually get a lot of information about parameters from the data, but at the same time, we know that there's a huge, the more parameters we have in a model, that introduces more degrees of freedom and more uncertainty about whether the model is correct. So this is all part of the model building process, though. And then at the end of the day, we need to perform simulations of the models that we can then compare to data. And we've talked a lot about already in the workshop, essentially a Brownian dynamics or particle-based stochastic dynamics simulations of biochemical models. I'm going to talk in this segment more about ways of simulating models that are well mixed or compartmental. So we're, in the reactor volume, we assume that the molecules are well mixed with each other. And we know for sure, those of you who are cell biologists know very well that the cell is not a well mixed bag of chemicals. But many processes that go on in cells can be adequately represented in that way, and some can't be. So we're still figuring out which ones can and can't be represented by well mixed assumptions.
So at one point, somewhere, maybe after lunch or something, I have one slide on the whole.
Yeah, the picture. Yeah.
The picture, and maybe we can have a discussion about that.
But an even more useful way to actually approach that problem, maybe you should put this up after, is if we look at something like Latka-Volterra, right? Because we can then look at how do each of these different, we skip this one, but how do each of these different approaches, what answers do they give us to that problem? And the PDE would be actually very interesting for Latka-Volterra as well. They're all going to give us different answers. Same interactions, same constants, different approaches to simulation, different representations of their interactions in space and in molecularity, I guess, is the discreteness, give it completely different dynamics. Completely different. So yeah, it's interesting. So that's to say. So I guess that's good motivation to say that this is actually important, right? Like, this part of the problem is important. But what we want to be able to do is separate the part, that's the molecules and interactions, the model, from the ways that we simulate the model, in order to be able to make controlled comparisons and to get an understanding of what parts are fundamental to this and what parts are fundamental to this. And in some ways, they're mixed together. So we're never going to get a complete separation. But we want to have as much control as possible when we make comparisons and understand what aspects are controlling. So OK, so a little bit through this process. I talked about this already. We have molecules that interact with various structured domains and sites of post-translational modification. We can represent those in a fairly concise way as structured molecules in our rule-based language, in BioNetGen in particular. The components of these molecules, the guys that are inside the parentheses that are shown in red here, represents potential sites of binding interactions between proteins. And they can represent different aspects. They can be both concrete aspects of proteins, so they're elements of their structure, domains, or short linear motifs. They could also represent generic properties of the protein, different conformational states, for example. Okay. And then certain components can take on any of the components in a protein, but when we make a model, typically certain sites can have post-translational modifications, such as phosphorylation taking place. Those are represented by states. So here the tilde notation indicates the state value of a component. And the fact that we have two tildes represents that this component can be either in the U state or the P state. So you could think of this as a type declaration for a protein. It says the molecule Fc epsilon R1 has A, B, and gamma 2 components. B can be in the state UP, and G2 can be in the state U or P. And similarly for 6 LY and AY domains. Yep. Right. You could have, and if you have a model where you had only one, I mean, maybe you anticipate that sometime you would have two, but not in this model. Yeah, it's possible.
The phosphorylated state would always be represented if the component is phosphorylate.
Right.
Phosphorylable will always
Right. You could argue, and based on what you just said, you could argue that this is kind of redundant. That if I have a tilde p, the implication is I had something else also. And in fact, yeah. But this is how we do it now, sort of. Yeah.
But if you had a bigger model that also needed to keep track of the energy metabolism in the cell and the phosphorylation tension, right, in the cytoplasm, you would need to keep track of the P as a species in its own right, not as just a throwaway token, but as something that.
Right. If you wanted to model phosphate balance, for example.
Yeah, exactly. So having it here.
That was.
Context isn't redundant.
Yeah. Yeah, I mean, there's an infinite number of ways to represent those, and the language is not saying you have to do it one way or another, although it's definitely helpful if there's consistency in the way that if you adopt conventions, at least that are internally consistent within your model. And ultimately, I think we would hope to have conventions that are shared by community to make models more easily transferable. But at this point, there aren't any. Okay. So the first step then in making more complex interactions is making bonds between molecules. So in the BioNetGen language, I already showed you an example of a rule in which a bond was formed. We represent the bond with this bang or exclamation mark notation to indicate that a given component on one molecule is linked to a given component on another molecule. And then the dot between molecules just indicates molecules that are part of the same complex. So
whether or not they have a bond. Oh, I see. So you're saying the IgE.
So here, yeah, the IgE is bound through one of its A sites to another, to the A site of the Fc epsilon R1 receptor. Okay. And this is a specific complex. So in a specific complex, we have the beta and gamma subunits which have states. They have to be in a specified state. To make this an instance of a given complex that could occur in the system, I have to know what the state of all of the components are. States of all the components are.
But so for this particular species, that's the correct way of writing that, say, for this thing. But for, say, some other imaginary species that I could think of. So in this one, you have IgE and FCRI that are in the same complex because of the dot, but they also have to be bonded to each other through that A bond, right? But I could imagine another species where I have IgE and FCRI or X and Y, right, in a complex. So I could have a dot between them, but they, which just means that they're in the same complex, but they wouldn't necessarily have to be bonded to each other, though, to be a member of the same complex.
Right. They don't, but they have to be. So to be a proper complex, to be there would have to be a path of bonds that connects them.
Right.
Okay.
If it's don't write
if it's so that's for it to be a hey Jose, what's the term that we use for a complex in an actual species? We have a name for that. I can't remember. I'm drawing a blank. Where every component component that takes a component state has a defined state, and all bonds are represented. A proper species, an instance, yeah. We have another name, but I can't. I'm drawing a blank on it still too. But anyway, for an instance, you have to have. There has to be a pattern, but a path.
Right.
For a pattern that we're going to use in a rule, there does not have to be.
Right.
But there are consequences to making disconnected patterns. In other words, if you say I need a molecule of R and a molecule of L in my complex, but I don't care how they're connected, that means I have to, when I'm figuring out if a given complex in my system satisfies the criteria to be a reactant, I have to check and see if there's a path between any R and L in any complex. Okay.
Why
Yeah.
can't you just check the...
I mean, you have to...
Why can't you just check the instance?
You have to
if
see...
if they're
Well, for every
present?
instance, there are some shorthand things you can do, but it's still computationally intensive, more intensive to deal with that problem. Yeah. Yeah. That gets into simulation algorithms, which we will get to, but...
Just esoteric.
Okay. So I think the one other point that's worth mentioning on this slide is that components that take states can also be bound to other components. So the beta subunit can be unphosphorylated in this case and bound to, we look for, here's the link, and then the terminus is the lin tyrosine kinase through its U domain. Okay, so this is a bond between the receptor and the lin. We see that the receptor here is unphosphorylated, so it's not true that binding and phosphorylation are always linked in the same way. Here's a case where the unphosphorylated state binds to a particular domain of the kinase, and then in the phosphorylated state, it will actually bind to a different one. Okay, and the last point here is a minor one, which is that you can have bonds between components in the same molecule. So in this particular kinase, the SH2 domain can be bound to the C-terminal tyrosine that's phosphorylated and form an inhibited complex that has low activity. Okay, so the rules look like this. We've seen examples of reaction rules before, and these are actually truncated rules where I haven't put the rate constants in. But I just want to show you how, some examples of more complex rules that have additional context. So here we have a dimer of IgE molecules that's represented pictorially like this, and I'm abstracting that as an IgE complex that has two A sites. These represent the Fc portions of the antibodies, for example, binding to the alpha subunit, extracellular subunit of the Fc epsilon R1 receptor, and all of the components are called A. So we see, just a pragmatic language aspect. The namespace of components is protected by the space of molecules that it appears in. So I can have two components on different molecules named the same thing, and I see I can have two components on the same molecule named the same thing. And as far as I know, BioNetGen is one of the few, is the only rule-based language that has that particular feature, that you can name components, that you can have identical names for components of molecules. And BioNetGen recognizes the symmetry of this situation. So these components are considered exactly symmetric for all purposes. So the fact that there are two binding sites called A means there are two sites on that protein that are going to behave identically. Okay, and that's going to create symmetry factors or multiplicity factors in reactions that I generate as a result of that property. And here I have both of them in this rule, and the net effect of the rule is to create a bond between one of them and the A subunit of the A component of the other receptor in the complex. And if you think about it, that actually breaks the symmetry here, which is relevant in the computational details of the procedure. But from the point of view of specification, it's completely equivalent to put the bond on this A or this A. I just put it on this one because it's closer to this one and make the bond easier to see. But there's the order of components in these rules does not matter, in the patterns does not matter at all for the purposes of how they behave in the system. The order of the reactants can matter under certain circumstances, but it generally does not. So here, I'm forming this bond, and so the reaction center is an A molecule of IgE and the A component of IgE and an A component of the receptor. And there's an additional context here, which is I require the IgE that's reacting has to have initially two free A sites. Okay, so that means only the unbound IgE dimer can bind to the receptor, and not one that's already bound. So I'm making this rule explicitly, the one for the doubly, the IgE dimer that has neither site bound, as opposed to this one, which has already one of the sites is bound and one of them is unbound. And we'll do that, and we'll actually make these reactions in the lab. Okay.
You said the order doesn't matter, so that means that one line represents the whole permutation set of these things with arbitrary ordering?
Right. For example, BioNation will generate 2 instances of this of the IgE binding reaction initially. So it will represent. It will make a reaction for binding to this first A molecule, this first A component, and then the one, or the second one, and then the first one. It will realize that those are symmetric and collapse them into a single reaction involving two species. And then the reaction rate will be doubled because there are 2 possible ways to do that. So
So it works the same way if A had state, say, unphosphorylated, phosphorylated, if we just write one rule for one of the A sites.
Right. So here we haven't mentioned what the state of A is. So if A had multiple states, this would apply equally to all of them. It doesn't care. Since we haven't referenced the internal state of A, it doesn't care about that. Neither does this rule care about the states of B and G. So this rule actually generates a large number of possible reactions because the IgE can bind to a receptor even if the receptor has been phosphorylated or is bound to another kinase, and so forth. So what's going on at the beta and gamma sites inside the cell doesn't affect this binding reaction. Of course, that's an assumption, right? That we've made in constructing the model.
So if you specify that the first A in IgE has a reaction where it can become phosphorylated, that's enough to specify for both A's?
Yes, absolutely. Yeah, if I had 50 of them, I would write one rule, and then that could make all 50 be phosphorylated. Don't try that one. There are some issues. So the symmetry is probably one of the most confusing aspects of the language for people. It's also confusing algorithmically, and the handling of it is not perfect, but it's pretty good. So you can do multi-site phosphorylation very easily. And that's a trick that you can use to make. To write one rule to handle phosphorylation at multiple sites, and it's clever. It will only, like if you had 4 sites, it would make 5 species, because you can have 0 sites phosphorylated, 1, 2, 3, or 4, but it wouldn't make 2 to the 4 species for those.
It's a big part of the trick, right? That makes it compact. Right. So you don't have to write down 28 million.
Yeah.
Yeah, I'm also just thinking, from a coding perspective, it's probably safer to just write out that, each A has different states, to actually write out all the rules.
If you had multiple A's, you mean? So this is you could get into there are philosophical discussions that can be had about this problem. Some people believe that there's, in protein biochemistry, we shouldn't have symmetry because, symmetry naturally, like in a polypeptide chain, you really don't get highly symmetric molecules. I've heard that argument be made. I don't agree. There are many, many cases in biochemistry where symmetry is quite useful. Just think of, what's it called, the hemoglobin example. So we exploit, but yeah, you should use it judiciously, I think is the best. You can definitely make confusing models by overusing symmetry properties, and you should. Yeah, but there at the same time there are cases where it is quite useful.
I would say that in a single
Yeah, you get
polypeptide,
yeah.
in a single chain of amino acid, there might not be symmetries, but in a complex.
Oh, there's no question about that.
And you form crystalline,
So,
geometric crystalline structures that definitely have symmetry.
So a case where that Tom would be very familiar with, where this could come up, and I just mention it because it goes to the issue of how do you represent different cases. So if you think of CAMK2, it exists as a dodecamer. So there are 12 subunits that form in two 6-membered rings. So there's a symmetry. There's 6 identical ones on the top and 6 identical ones on the bottom. They're all the same, effectively. But their relationship, because of symmetry, their relationships are not all the same. But so there are different ways you could try to represent that symmetry. Well, one way you could do is to make one big molecule that has 12 components and call them all. Say there's a tyrosine or a threonine that gets phosphorylated. And you just call that, T1, and you could make 12 of them. And then whatever, so every T1 site of all of those different CAMK2s would be represented as a distinct component. And then phosphorylation would occur at one site. You'd write one rule for that, and you would have all of them. If you wanted to consider the possibility that phosphorylation of one affects the phosphorylation of the other, you would have one pair, one rule for the pair, and so forth. And that would make your model a lot simpler. Another equivalent way to do it, at least at the beginning, would be to make one CAMK2 molecule with, say, your T1 site, and provide another site to link those CAMK2s together and construct the dodecamer by hand, in your code, or by script in a code, and you could even, you could make 2 hexameric rings linked together, or you could make, a 12-mer hooked together as a chain. That would obviously have slightly different symmetry properties than the real CAMK2. These are all choices that you can make in building up your model. But the main one is whether to represent each subunit, each separate polypeptide in the complex as a component or as a separate molecule linked together. And so here, for example, there are examples of both kinds of things going on. So here in Lin, we represent the components represent domains of the Lin tyrosine kinase. At this level, at the receptor level, the components represent different subunits of the multi-subunit receptor. And you could argue, well, you're not consistent, and you could, that's just a choice that the modeler is making at this point, and how to compose these structures. And when so one of the things that's very interesting to think about doing is all of this is basically these rule sets are rules for making polymers, and we've made simple models, for example, of DNA replication based on a rule-based approach, or models of translation and so forth. And it's kind of fun to play with those. But, OK, we're digressing a little bit too much, so let me just bring myself back onto focus here. I just wanted to show an example of a component state change. So binding and unbinding and component state change represent, well, a large fraction of the kinds of rules that you're likely to write. The other kinds of rules you would write would be synthesis and degradation. We'll talk about those a little bit later on in the lab, where you make a molecule or you break a molecule, destroy a molecule. But for many purposes, these are the 2 main kinds of reactions you're going to be making. Here we're changing this. The only thing that this rule does is change the state of the receptor beta subunit from unphosphorylated to phosphorylated, but it has all this other context associated with it. So here, it's the Lin that's associated with a receptor, an adjacent receptor in the complex that's causing the phosphorylation at this site. So this is all context for a single phosphorylation reaction to take place. And the pictogram, the biophysical representation of how this interaction is occurring is shown here. So we have a Lin that's associated with another receptor in the complex, phosphorylating the adjacent phosphorylation sites on the adjacent receptor. And this actually is experimentally has been shown to be the dominant process in this system. So and one of the reasons that we really developed BioNetGen was to be able to represent complex biochemical interactions within a complex in as simple a way as we could think of. Okay. So this is like an example of one of the more complicated kinds of rules you would see in a system.
You could draw that diagram.
This one.
Yeah, that one, that makes it look like that's what's happening. But another interpretation is
Yeah.
it's the state and that phosphorylation just happened.
Right, right. That's one weakness. We don't have a way. So if you wanted to represent the full mechanism, you would have to have the kinase domain of the Lin molecule here, binding transiently, for example, to the phosphorylation site here, and then undergoing say an irreversible reaction to add the phosphate and coming off. We're actually going to do a Michaelis-Menten example like that in the lab this afternoon. I'm thinking that what's going to happen is we'll take a break, and we'll come back, and we'll have the short presentations, and that'll take us up to lunch, and then we'll do the lab activities in the afternoon. So hopefully does that make sense, Jose? Okay. Since it's going a little bit slower than I thought it would.
What you just said, though, you could write it that way.
I could, yeah, I could. That would be 2 or 3 rules, depending on how I think about it. And the disadvantage of writing that, the advantage of writing that way is clarity. Its mechanism is absolutely clear. Disadvantage of writing it that way is then I have all the extra states of those transition complexes that are associated with that.
The rate constants.
Right, although if I think of it in terms of Michaelis-Menten, I'm going to have 3 constants where I would have had 2 before, so not so big of a... actually, no, it's really 1. Here, I represent this as unimolecular. The big advantage of what you're talking about, of explicitly representing the interactions with the kinase, is that this is absolutely a saturated reaction. That when these guys are unphosphorylated, so the kinase is sitting out here in space. It's very close to these substrates. As long as it's not sterically constrained, this is going to happen. This reaction rate is going to happen at the rate that's limited by the catalytic rate of the process. So these guys are going to get very rapidly phosphorylated, but then once that happens, if they're phosphorylated, then this kinase is sort of free to do other things possibly. And so, how does that fact affect the behavior of these reactions in a complex network? It could. Yeah, it hasn't been very adequately explored for reasons that are probably starting to become obvious. You would have to write down a lot of equations in order to figure that out if you don't have BioNetGen. Okay, so I've already gone through this before, so this should just review basically and going back over the reactions that I showed, the rules that I showed. So here's our IgE rule again. I've highlighted the center of the reaction here. We have the reactants, products, and our rate law. And how does the rule operate then? So if we talk about specific instances of molecules in our system, so initially, if we think of a cell at rest where we've added IgE to the supernatant around the suspension of cells or on the top of a layer of cells growing on a plate, we have our free IgE dimers, and those are in solution, and we have our receptors that are unbound and unphosphorylated. And this rule now is going to generate the possibility of a reaction occurring between these two, because it says that if I have free IgE dimer, so both A sites are unbound, the free receptor, the A site is unbound, then these patterns of the rule match on to these particular species, and now can create a reaction based on that. Another example of a receptor state that could be applied by this rule is a receptor that's unbound but has been phosphorylated somehow previously prior to this encounter with the ligand. So again, this is an example of our don't write, don't care. This rule can generate many possible reactions depending on the different possible states of the receptor. Okay, and then what's shown here on the right-hand side are the specific instances of reactions that are generated by this rule. So here we're making the bond, we're forming the IgE dimer bound to an unphosphorylated receptor, and here it's bound to a phosphorylated receptor. And the key point, and go back to our parameter discussion, the reactions generated by this rule both inherit the same rate constants that were associated with the rule. So every possible reaction that's generated by a rule inherits the same rate constants. If I want to hedge just a little bit, I could say the same rate constant functions, because these can be functions of anything in the system. It's possible for these to be functions of the specific reactants involved in this rule. And so in that way, these functions could apply differently depending on the specific reactions that are involved. But that's a very advanced usage scenario at this point. Yep.
Would it be possible on the product side to write different, specify different components that are affected by the reaction than you care about?
No. If you want to do something to a component, you have to specify its state on the left-hand side.
And what if you don't care what its state is on the left?
That you still have to specify it. We allowed in earlier version.
Lots of possible.
Yeah, there are definitely some cases where that's a bit of a nuisance. No, it has come up. That issue has come up. But current spec requires that that everything that you're going to change has to change from a specified state. You can't change something from an unspecified state. To avoid, it's to prevent you from making certain kinds of errors.
But in this case, do you have to specify on the left-hand side both of the A?
If you didn't, you don't have to. You don't have to. But then you would get reactions involving an IgE that's bound already. So let's just say we wrote the rule and we deleted this A. Okay. So what would happen is we could apply that rule would still apply to this pair, right? And we would make this complex as a product. Okay. But now this guy still has a free A, so it can also be a reactant. So we would also match, and we would make then through the same rule, we'd add another receptor, and we could make the dimer, which is fine. Except that, as we'll see in the lab example this afternoon, typically if we have a situation where if you wanted to make dimerization, I would argue that's not the ideal way to do it, because if our first receptor is here, and then we bind a ligand molecule that has two sites for binding the receptor. For this first reaction, the ligand is binding from solution. So its effective concentration is determined by the volume of this extracellular space. For the second reaction, this ligand site is now bound to this ligand is now bound to the surface. So its effective volume is greatly reduced to basically just this shell around the cell that's swept out by this diffusing the ligand receptor complex diffusing on the surface of the cell. So the volume here is greatly reduced compared to the extracellular volume. And that means that the concentration is increased, and the effective rate constants governing this second reaction are much higher. So the K plus 2 is much greater than the K plus 1. So for the first reaction, we have a K plus 1 constant governing the ligand receptor association. And for the second one, we should have a higher constant, essentially. And so that's why, like when we make the model of the system, we separate the two rules. We write the one for the free ligand binding from solution, and then we write the one separately for the one that's already bound. Yeah.
So this is a case where thinking about space is important, right?
Thinking about compartmental models already, so after the first binding reaction, we should already be considering
Yeah. In compartmental bioNetGen language, which we'll talk about again, this afternoon probably, you can set it up in such a way that you could write one rule and it would automatically take this reduction of volume into consideration. But multiple things happen when the ligand is bound to the surface. The diffusion constant of the receptor is the diffusion of the receptor is limited to 2D. Its diffusion constant may be smaller than, and it may effectively be, the reactions may become diffusion limited, so that it's not always the best to have just one, to just consider it a reduction of volume effect. So.
And in thinking about how you would write this in the
Yeah.
And they would have, and you would write the diffusion constant of a volume molecule, the diffusion constant of a surface molecule. It would all just happen, just the dimensionality and the change of dimensionality of diffusion, the change of concentration from concentration to density, and the change of diffusion constant would all happen by writing it down with space in mind when you did it.
Okay. So we talked already about the concepts of center and context, so this is really just a review of that idea. The center is the part that's changed, the context is the part that's required but doesn't change. And we see center and context in these two reactions are highlighted. So to make a long story short, a rule-based model is a set of molecules and reaction rules. We're going to go through some examples of these, and there are many examples that are provided on the wiki in addition that you can go through later on your own as your interest dictates. What do we do now with the model where we've specified these molecules and interactions? So this is the model of the system. How do we simulate that system? And so the last part of the presentation this morning is going to be about how do we simulate rule-based models. And there are two basic ways that we perform simulations. And the standard one that most people will have been exposed to is what we call the indirect method. So we have our model, our set of objects and rules. We generate a reaction network, all the possible species and reactions that are implied by this set of molecules and rules. So we generate a network, and we get a reaction network, set of elementary mass action reactions that we've seen. And then we can simulate that in various ways using either converting that either to a set of ordinary differential equations. We could simulate that as a set of partial differential equations where we considered diffusion in space. We could simulate this as a stochastic using stochastic simulation methods, and we'll do various kinds of perform various kinds of simulations of generated networks in the lab. We can also take this reaction network and export it to MATLAB, to SBML, to MDL, and run it in MCell, for example. We have to combine that with the geometry. And I don't know if you guys did you guys do any of that in the lab yesterday? I don't know how far you got. Did you do an SBML model in the lab? Okay. Okay. So we'll see that this afternoon. So this is a fine way to do things as long as the model is not too complicated. So you can generate reaction networks with thousands or hundreds of thousands of species and reactions, but there comes a point where you can no longer generate the network. And in that case, we can use so-called direct methods of simulation. And here, instead of simulating a set of possible species and reactions, we simulate only the ones that arise in a discrete collection of molecules. So we start with a certain set of molecules. We have rules that can create bonds and change their component states, and those update the state of our system continually as we perform a simulation. It's very much like a molecular dynamics simulation. We have a set of proteins in a box, and they interact with each other through some potential. We know what the forces are, and we can track their movements in time over the course of a simulation using solving the F equals ma Newton's equations, for example. Here we're solving chemical kinetics equations. And I'll talk a little bit more about this too. So the basic methods are indicated in a couple of slides here. So for indirect methods, we have an example of a set of proteins that bind to a scaffold. The scaffold is called S. The proteins are A, B, and C. And so our scaffold is here. It binds A, B, and C. And we have here just three separate rules. So there's going to be no cooperativity in the binding in the scaffold initially, in our initial assumption. So we have three rules that specify that there's no context in any of these. They just say that A binds to the A site, B binds to the B site, and C binds to the C site of this scaffold. And then that generates a reaction network with the following reactions. So the A binding reactions are shown here, the B ones are shown here, and C ones are shown here. There's a total of 12. There are 11 possible species, which are indicated by shorthand here. And that's it. So at the end of the day, we have a reaction network with these 11 species and 12 reactions. And we could we could write down differential equations now for each of the species. And we could solve those either numerically or, well, probably only numerically by simulation on the computer. OK. And given that reaction network, we could also perform a stochastic simulation using Gillespie algorithm, for example. And we could compare the dynamics of the stochastic simulation with those of the differential equation, for example. So does everybody understand what I mean when I say how we would translate this reaction network into a set of differential equations, for example? Or raise your hand if that doesn't make sense to you. You don't know what that means.
What does it mean? S plus A, S, S.
Yeah. So that's just a shorthand representation of the species S. So S means unbound to A, B, or C, to the molecule A, which is not bound to anything. So instead of writing out the full BioNetGen code for all of this, we've used an abbreviation here. So I mean, as an example, I'll just do it for completeness. I just erased I'm sorry about that. So an example would be that, so if the differential equation for A would be the set of all of the reactions in which A can participate, which either produce or degrade A, would contribute to this. So we would have, for example, K1, a term K1 times S, concentration of S times concentration of A from the first reaction, and it would be a minus, right? Good. And then the reverse of that would be plus K minus 1. I think that's what you use here. He calls it K1 minus. OK. This is K1 plus, K1 minus, times the concentration of the complex A. Typically, we would write the brackets around here to denote concentration. So, and so forth, right? For each of these reactions is going to contribute a term in the forward direction and the reverse direction to A. And so there will be 8 total terms for A, 8 for B, and 8 for C, and 24 total ones for S, because all of those reactions affect the concentration of, oh, actually no, S containing species, but not S itself. S has only 3. So we don't need to get too involved in the details of this, but just to know that if we know the reactions, we know the differential equations. If we know the reactions, we can also perform a stochastic simulation. In a stochastic simulation, every one of these arrows corresponds to a possible process that can occur at a given time in the system. I use a Monte Carlo procedure to select which one occurs at the given time. Say I pick a particular reaction to fire. That means that I drop the number of SC complexes in my system by 1 and B complexes in the system, and increase the number of SBC complexes. I think I might even have this. No, I don't in this deck.
And if you did it in MCell, you would just write down the 12 reactions.
Right, it's the same. So when we export a model from BioNetGen to MCell, we're exporting it in this format.
And you wrote that there's 12 reactions, but there's actually 24 reactions.
Yeah, there's 12 bidirectional reactions or 24 unidirectional reactions.
Right.
Yeah. And BioNetGen would report, if you made this model in BioNetGen, it would report 24 reactions.
So when we specify the differential equations, we also need to specify the starting points, the initial conditions.
Yes, yes. The initial concentrations you will need to specify. And those nuts and bolts will be in when we do the tutorial. We'll see it exactly. Yeah, just it's exactly the same. We already did the basics in the MCell tutorial yesterday. It's the same thing. You have, once you reduce it to this form, it's exactly the same as doing a model in MCell. So the thing that's the method of simulation that's different between BioNet and MCell, still, although we're working on implementing it into MCell, and they're never mind, that's good enough, is what we call direct methods, where we simulate a collection of molecules directly without worrying about all the possible reactions in species, but only the ones that we encounter at a given time. And so the idea is illustrated here. So we have our different reaction rules. At a given time, we have a configuration of the system. We have our scaffold molecules and our binding partners, A, B, and C. And there's a distribution of molecules that are bound and not bound in all the various states. But those are the only ones that I have to consider. So at any given time, I need to know the number of species of each corresponding to each rule that are currently existing in the system. So for example, there are currently 6 scaffold sites, 6 scaffold molecules with a free A site in the system. I think that's correct. Should be 1, 2, 3, 4, 5, 6. And there are currently 4 free A molecules. And so the total number of reactant combinations in the system is 24. And the total rate is going to be 24 times the reaction rate constant, k plus 1. And so for each rule, we need to compute the total probability, the total rate of each reaction firing. And in the stochastic simulation algorithm, the probability of a reaction firing is simply proportional to its rate. So we're gonna select over one of these possible rules to fire over all the possible rules, and then we'll update the system accordingly. So we have the total rate of all reactions firing in the system is given by this total propensity. In the Gillespie algorithm, that determines the time or the distribution of probability of reaction occurring at the next time. And then
Assumes that everything is so close together that every molecule is everywhere.
yeah, this is a well mixed. So assumes well mixed, so that the reaction rates are completely determined by the total concentrations of the species that are involved.
And so to be explicit about it, if it were a spatial simulation, that's saying that the diffusion constant is infinite, right?
Yeah, you can imagine quite easily how you could, like, if we thought of if this represented a two-dimensional distribution of our complexes, you could easily imagine that you could assume local only mixing in a local scale, so that you would have domains and you would compute the concentrations on domains, and that's the basis for one of the standard ways to perform stochastic simulations of a spatial system.
And this kind of enumeration where you're counting up the numbers of each one, that's really the chemical master equation.
Right.
Where now you're putting specific numbers for the quantities of each of the possible species.
So is there any biologically relevant case where this would actually be really realistic, or?
It's yeah. I mean, we use it all the time. So
It's also what you do when you write down an ODE.
yeah, I mean, this is same. It's the same as any differential.
You do
So
that all the time.
for the molecules, for the reactions that occur in the cytoplasmic space, for example, in the volume, this is pretty good. It's a little bit more dicey for reactions that occur principally at the membrane, involving only membrane complexes. But many of if we think about, like, if we imagine that our S molecule in this example is a membrane-bound receptor or complex, and the A, B, and C are molecules that are cytoplasmic that are binding to that complex, then we would expect that this would be a pretty reasonable way of describing the kinetics of complex formation. If we wanted to consider dimerization of membrane-bound complexes, it gets a little bit more unclear about when that's going to break down, because we know that there is going to be spatial organization in the membrane of complexes. However, I mean, nobody has a surefire way to simulate those things at this point.
So another way of gaining intuition about it is to think about, well, when we play around with these didn't talk about it. We didn't talk about it yesterday in the last little pair, but the first case that we had that had all the patches of stuff happening, that was the purely diffusion limited case where the reactions happened instantaneously fast, and it was diffusion that was slow relative to the reactions.
So this, we're going to be doing some hands-on rule-based modeling in the lab, and this just gives you a snapshot of the interface that we've developed for building and analyzing and simulating rule-based models. So we'll be working with RuleBender this afternoon. And just to finish up, to summarize, the procedure, the process of rule-based modeling looks something like this. So we build our objects and rules to describe molecular interactions. We then pass those to BioNetGen. We can make a reaction network out of them and solve them using ODEs or the Gillespie SSA, stochastic simulation. So these are well-mixed simulation methods, all of these actually. Or we can directly simulate our set of rules and molecules using the network-free simulator. But in the end, the output looks similar. We have time courses of various variables that we're interested in tracking in the system, such as, for example, the amount of SOS recruitment to the membrane in our earlier model. And we can see their different model assumptions and structures give different behaviors of the observables of interest. And we're interested in the modeling process to explain how those differences can arise. And I've given links to the various tools. The universe of things that can be done with BioNetGen is ever expanding at this point. So there are a lot of different tools that can use BioNetGen language models. And one of them, importantly, is what we're going to be working on with the lab. We can export models, reaction network models, into MCell and perform simulations considering not only the molecular complexity, but also now spatially realistic geometries. And we'll do some of that this afternoon. What else do I want to say? The dashed line, yeah, a direct connection to specify molecules and interactions and then perform a simulation directly without actually generating a network. There are non-published or features.
A paper just came out.
Oh, on
doing non-published.
Okay.
Basically non-published thing. So we haven't talked about it yet, and there's no way to specify it now in
Cell Blender, yeah.
But there is an experimental, but now published syntax for doing rule-based directly in MCell, but it doesn't use BioNetGen language. It's another thing, and it's not as elegant and complete as the BioNetGen language. But if anyone has a model that they need to run today, or in the near future, that suffers from combinatorial complexity and needs to do it in space, there is a way to do it in a limited way in MCell now. If anyone has a model like that, let us know, and we can help you with setting that up.
Yeah.
There was a paper in PLOS CompBio techniques, or it's a technique.
Yeah, I can't remember what it's called.
It's an online.
It's kind of a review, right?
Yeah. Review where we explain how it works.
So I just wanted to thank people who've contributed to BioNetGen development from my lab and others over the years, and thank you guys for being a great audience. It's been fun talking about this stuff. So we can take a break, and we'll come back and have some presentations on recent applications of rule-based modeling.